Okay, good morning, everybody. We will get started. We left off a little bit, almost halfway into the chapter, with some mutations, and there are lots of different potentials that can happen when we think about the genetic code, the DNA getting altered, all right? This initial stuff that we want to start off thinking about are what we call point mutations. An individual base gets altered. Whether it causes an issue or not, we will see, all right? Oftentimes, when we think about mutations, we think about them causing problems where the proteins don't get produced like they should. Sometimes we don't recognize it at all. And occasionally, those mutations might actually confer something that allows the protein, whatever it might be, the structure that we are producing maybe has some sort of a benefit that we didn't recognize before. So it could be beneficial, but oftentimes it's not, all right? So just depends on what's truly getting altered in this process. So again, like I said, a point mutation is just a single base that gets altered, all right? And this first example is a silent mutation. So we're altering the sequence, and we can see in red the base that gets changed. It could be nice to see that amino acid table right over here so we could make some observations of what's really happening, but we can see what it's coding for, all right? So as we see this, as we move through, this adenine gets mutated, and in turn, a different nucleotide, and we'll show some potentials for how this might happen in a second. What was an adenine in the normal sequence, wild type, what should be happening, changes to a thymine, all right? But as it turns out, as we're reading these three letters, this situation ends up coding for the same amino acid. They both are coding for valine, all right? So in this circumstance, although it's different, it doesn't matter. And we call that a silent mutation. We showed in that amino acid table some other redundancy where there are handfuls, potentially handfuls of ways that you might code for the same amino acid. And that in turn, we could think of as some of these silent sorts of mutations, silent shifts in the nucleotide sequence, but doesn't matter because we're still making the same stuff, all right? That's all fine and good. But oftentimes it's not something like that. So a missense, now it's something, you know, if we're thinking three-letter words as we're moving through here, these codons making the respective amino acids as we see here. Now, what should have been a proline ends up being a threonine, right? Somebody completely different. And there's all sorts of differences with the respective amino acids. Some of them have some positive, some have some negative charges, some are acidic, some are basic. As the amino acid sequences are generated, they twist and fold and coil up on themselves. And potentially this new amino acid that's thrown into the mix causes this protein to not fold like it truly should, all right? And in turn, losing that function, all right? Oftentimes, right? But like I said, sometimes maybe these shifts may cause something slightly different. Maybe the way it's folded now has slightly better function, gives us a sort of an advantage that we maybe didn't recognize before, right? But typically, like I said, it's going to be bad news. The nonsense, not that missense doesn't seem nonsensical as well. Nonsense mutations are when we wind up with the substitution that generates a stop codon. So it could be mid-sentence, mid-sequence of this amino acid chain, where all of a sudden you slap a stop code on in there. Now, what should have had 150 amino acids linked together only has 23 or something like that, right? And in turn, you're not building whatever it is you thought you were building. It's not happening, all right? Because you stopped midstream, all right? The next bit, not the top two, or these two aren't potentially setting up situations for bad news. Nonsense definitely is. Insertions and deletions, this opens up all sorts of different potentials where we see the sequence at the point of either insertion or deletion. We oftentimes shorten it and slam them together and call them indels for insertions and deletions. Either one can be causing some issues, all right? So here we've got a deletion. We could flip it around and slap a couple nucleotides in there for an insertion, all right? Doesn't matter. Once this happens, you've removed a couple bases or you've added a couple bases, you shift the reading frame. So you've either extended or deleted part of that sequence, and we're reading 3 letters as we move through here. So once this happens, we've shifted everything over, and now from this point, our first amino acid truly that we're seeing right here, everybody past that is completely different, and maybe we wind up with the stop code on somewhere in there as well, but we're not building anything that we thought we should be building from the initial sequence. All right, different, all right. And as this reading frame gets shifted, really when we think about a lot of point mutations, these are typically more of the bad news situation because you've got bigger alterations than just an individual amino acid that might be varied. Everybody else is different from that point on, typically, all right? So mutations do have some potentials to confer some advantages. And this image, as we're looking at prevalence of HIV from a global perspective, there are some areas where the prevalence is really pretty low. And as we look at some of this with a little bit of Europe, I think is really the initial bit that the text talks about some details. There are some individuals that technically have a leg up with some things. And we can look back to really ancient times when we think about all sorts of, nasty pathogens, some of which still plaguing us. The plague, bubonic plague that we saw massive people dying. There were some that technically didn't. Maybe seeing some of the family members die, but some of these people didn't, right? And it makes you wonder what's truly happening, right? And through some mutations that ultimately alter some of our immune cells. T cells are what HIV technically wants to be targeting. And those virus can link up to receptors on those cells. And if everything works properly, the virus is brought to the inside of those cells and it infects it, and the process of replication starts. If your cells don't have the right receptor on there, the virus can't dock, can't make contact with those cells, and can't be brought in, all right? So there are some that have this mutation that in turn confers immunity, if you will, against numerous things, HIV being one of them, all right? And the text goes through and talks some details about this. I remember watching a documentary, this was years ago, that was really talking the same stuff. And they were looking at, HIV, really, all right? But thinking, as I said, in Europe, this is where we recognize some of it, and plague was huge, back in the day, in these areas. and it's got a lot of history. You can go there and, see things from centuries ago, and potentially cemeteries from centuries ago, which is, as we think in the United States, we don't go back that far, right? And in turn, it opens up potentials for doing some studies, right? People in the US, there are, they may go and look at, and they've definitely done this before, looking at cemeteries and looking at individuals. You may see an older portion of the cemetery, and all of the sudden there's a boatload of people from 1918, 1919, where there's all of a sudden a lot of tombstones. Lots of these people, we may speculate, could have died from the Spanish flu, swine flu, really, the big pandemic in 1918, 1919. Century later, here we are with a different pandemic, right? Looking at some of that stuff, all right? So they go thinking this stuff to Europe, and they start looking at some graves, and they can see families that look as if we've got quite a bit of people dying from the plague, right? And then they looked up some of these names. There are still some of these family names still living in these areas, right? So some of these people seems as if maybe some of their family lived during plague times and survived. They're still here, right? So they start looking at some of these people and extracting some DNA and doing some research, and come to find out some of these people have this mutation that potentially conferred super immunity, gave them a leg up, and they were able to survive things like bubonic plague. Smallpox might be another potential where they could have survived something like that. And as it turns out, seems as if HIV is another one. All right. The documentary that I watched, they showed this gentleman that lived in the US. He had he was a homosexual in California right at the height of HIV first being recognized, early 1980s, right? He in turn had numerous partners, right? He also noticed a little bit later on in time, lots of these friends and partners of his started getting sick. Lots of people started getting sick, and he started seeing all sorts of these individuals dying from AIDS. And he knew in the back of his mind, it's happening to me, right? It must be, right? But it didn't. And in turn, they tested him as well, and he was immune, right? He had these same sorts of mutations that they were recognizing in some of these individuals from Europe that they were speculating lent to their ability to survive the plague. Crazy stuff, right? And this was right before class started. I was flipping through the news the littlest bit and saw, and I've heard some details before about some individuals, potentially some bone marrow transplants, other opportunities maybe, but bone marrow is really kind of the kicker, where an individual could potentially be cured if that person that donated the bone marrow had the specific mutation that in turn allowed them to not allow those T cells to get infected. Someone that had HIV getting the transplant now has T cells that in turn can't be targeted, right? That's pretty impressive, right? So mutations, and in these instances, technically a good thing, right? Some of those people, as they were, looking at some of the details, there are people that seem like their immune response is super heightened, and in turn may say, I don't recognize ever being sick, right? Does that mean that you're one of these mutants that can't get ill? I wouldn't test it, right? But maybe, right? Maybe you are. Okay, so there's a lot of different things that can cause the DNA to get damaged, that could cause potential changes in the nucleotide sequence. Lots of things that you potentially may come in contact with that could cause these problems, right? Hopefully you're not coming in contact. So a couple of things here that we're looking at, things that mimic one nucleotide and in turn cause problems or disruptions, right? 2-aminopurine nucleoside looks like the adenine, right? But we've got issues, right? And in turn, and then we've got another one down here with the guanosine, where it comes in and doesn't behave properly, and we get different base pairing, right? So it's the aminopurine that we would think we should be looking at if we're adenine pairing with thymine, right? But it pairs with cytosine instead, right? Converting the AT base pair to a GC base pair after a couple of rounds of replication. We're not saying what we should have been saying, right? We've mutated. It's different, right? And that's ultimately the situation to lend to one of those point mutations, right? Nitrous acid is another type of mutagen. A mutagen is a substance, an agent that can cause some sort of damage to the DNA, right? So it modifies nucleosides like cytosines and causes them to produce some uracils instead when we're thinking linking up and going through the process of transcription, right? So that in turn, after some rounds of replicating in the DNA, we see it shifting the opposite way. GCs now become Ts and As, right? There's some other molecules or other substances, molecules that don't cause the bases to flip-flop around, if you will, or shift, but it causes the sequence of DNA itself to get altered, right? So intercalating agents, they get into something, they get into the nucleotides and basically cause some space in between the nucleotides that should be right side by side as our polymerase is going through reading stuff, whether we be trying to make mRNA or copying the DNA. Now all of a sudden the bases aren't side by side, and it causes problems with the reading of that. So here we can see this through some replication, a deletion that winds up occurring, or potentially insertions as we make this little skip going through there, and sequences get altered. That's what we're looking at here with the other strands that are no longer what we started with, right? So acridine is an agent that can get in and intercalate. Ethidium bromide is another molecule that is an intercalating agent. And when we think about molecular biology, we use ethidium bromide to visualize the DNA when we're running it out in a gel, because it gets in there and will show up, all right? But getting it on us can cause issues. It's a mutagenic agent and it could damage our own DNA, all right? So you want to be very cautious when you're working with those things, and you glove up, right? You oftentimes may see a researcher's lab where they're doing stuff like that, working with ethidium bromide or some other agents. And usually the benchtop where that stuff happens is denoted like taped off or something. So you know, this is an area that you don't touch, right? You don't roll in and sit your coffee down, right? And think you're good to go, right? Because you potentially contaminated yourself, right? So caution, right? Radiation, another potential that can lend to alteration and damage in our nucleotides. And it might be X-ray or gamma rays that can cause this damage, right? Breaking the backbone of the nucleotides, causing these molecules, the nucleotides, to fragment. And this would be some ionizing. Non-ionizing stuff like UV light, the sunburn makers, right? Ultraviolet light causes damage a little bit differently, all right? It's not breaking the covalent bonds, all right, that make the backbone. It breaks the hydrogen bonds in between the individual nucleotides. And specifically when we get to adenines and thymines and they're together on that strand, like we see here, two A's on one side, two T's together on the opposite. It breaks those bonds between the adenines and the thymines, and it causes a bond to form between the two thymines. This bond we call a thymine dimer, and it's not, as these guys are linked together, it's not a hydrogen bond like the A's and T's as they're linked together. It's a covalent bond fusing the two thymines together. It's a problem, all right? Even if they were hydrogen bonding, it would technically be a problem. But this is a strong bond. This is damage to the DNA that's harder to deal with, all right? But it's not impossible, all right? There are some DNA repair mechanisms that can recognize issues like this. They can recognize some issues like the breaking as well. If it's a double strand break, that might be a bigger problem than a single, all right? But these thymine dimers can get recognized and can get corrected, all right? Lots of cells have the ability to deal with this. Our skin cells have the ability to deal with this, all right? But it's not 100% effective, right? Skin cancers are an issue. And if all of this functioned properly, well, we technically wouldn't be concerned about that. And sunburns, who cares, right? But technically you should care, right? Because as we get lots of this damage, sometimes it winds up being irreparable, right? So that's one of the repair mechanisms, recognizing these thymine dimers. So when they covalently bond like this, it causes that strand that they bonded on, causes it to not be pairing properly, and it's almost like a little speed bump in the road, all right? You know there's a problem, right? Maybe we could think of it more, you know, wintertime, kind of like a pothole, right? It's a problem, right? So there are, with this excision repair, we can see, well, two different ways, all right? This first bit with the excision repair, a nuclease, an enzyme that recognizes we've got an issue, all right? And it's really a polymerase, DNA polymerase 1, that's doing some proofreading, all right? It's the molecule that goes in and chews the RNA primers off as we're replicating our DNA, and it puts new nucleotide, DNA nucleotides in there. And as it's doing other surveillance, it's looking for stuff like this, the little speed bumps. And when it sees that stuff, it doesn't just fix the speed bump, all right? It fixes by chewing out sections on either side of it and laying down all new structure. So you got a nice smooth transition, all right? And technically you need a little bit of room in there for all of this to happen. So these enzymes, the nuclease, chews off a handful of bases on either end, and in turn, we replace it with the proper sequence, and now we hydrogen bond properly. If we get more UV exposure, we got the potential for more of this to occur. And if there's boatloads of it, well, potentially our excision repair doesn't work like it truly should, right? Photolyase is another type of repair mechanism, and it is activated through light photo activation. And this, not ultraviolet, it's visible light, all right? And it in turn causes more reactions. Enzyme photolyase does a similar idea, but it's not going in and chewing off this stuff. It's breaking the covalent bond. keeping the same bases there, just breaks those bonds and then they're allowed to re-hydrogen bond. Both of these processes can be happening together, all right? So this technique that we're looking at, we used to do in these labs that you guys are taking, and we've modified it the littlest bit, all right? You're still gonna do something sort of like this, all right? But it's a little bit different, all right? We've also transitioned and started using some different bacteria in this mix to show a little bit of differences with it, all right? But what we've got here, this replica plating, there's a disc right here that's some velvet that's going to be your stamper, gets basically hooked onto. So you've got this nice little stamper, and now we're looking for some sort of damage, oxytrophic mutants. So these would be something that now requires something that the wild type of bacteria wouldn't need, right? So a vitamin, a nutrient, something like that, that they can't make on their own, but the wild type, the non-mutated type, they can live all fine and good without it because they can synthesize it on their own, all right? So we would use ultraviolet light to cause the damage and to create the mutants, all right? These oxytrophic mutants. And here we need histidine in the media in order for them to grow, all right? So we've got them initially on this medium, all right? And this one, it's got histidine, all right? We could expose them to the UV light. We could do whatever it is. We've mutated them, all right? And we've got with our replica plating, and you can kind of see right here, they've marked the plate. You would have a mark, you would have something on your stamper. So you know, when I stamped this on my initial plate, which also has a mark on it, we don't see it, but there's a mark someplace on there. I stamp it there, and then I put it onto these other plates. So they're all basically like 12 o'clock, right? Straight up. That way, when I go back and look at this plate and make comparisons on these plates, I could say these colonies, right, that we're all on here they are, right? Or here it's not, right? So this medium doesn't have the histidine, right? So now only bacteria that are not mutated would be growing on this plate, right? Because they can make histidine on their own. But this little colony right here, we can see it up here on the plate that's got histidine, but the plate that doesn't, it's not growing, right? So I could go back to this plate, or I could potentially go up to my one up here, right? And see that same colony because I've marked these plates and I know this bit and that bit's the exact same colony, right? And I could pick it off and know that's my mutant, right? Whatever it might be, right? So this wasn't exactly the same lab that we did. We did the same replica plating techniques, but it was a little bit different, right? We worked with a bacteria called Serratia marcescens. You've maybe worked with this one in the lab, or you will be. So Serratia it's an environmental bacterium. When it's growing happy, if you will, it produces colonies that have a pinkish sort of pigment in the colony, right? And we would grow these bacteria and we would do a similar thing where we would have a plate that's got good nutrients and one that's got basically lacking nutrients. So it's the same idea with like the histidine or lacking histidine, right? So full nutrient agar plate, it's got all of the good nutrients in there. And then a minimal media plate is lacking a bunch of those nutrients that the nutrient agar plate had, all right? So we'd stamp both of them and we'd grow these things up, all right? We expose the Serratia to some UV light to cause some mutations, and then we would see some of those colonies growing on the nutrient agar plate were not growing on that minimal media plate. And you could do the same thing and look at it and say, oh, this one's mutated, all right? But that's a boatload of little velvet cloths. It's a lot of work to deal with all of that stuff, to clean it, to re-sterilize it, to go through all of those steps. We did it and it was kind of interesting, but it was really kind of a lot of work to make all of these things happen, and boatloads of plates, right? As we started doing these experiments, we made some observations that started us thinking, maybe we could do it a little different, all right? Maybe we don't need the velvet, right? Serratia, like I said, is pink when it's happy. And we recognized that these bacteria, when we exposed them to the UV light, and then they're growing up on the new plates after we've done our stamping, some of those Serratia no longer are pink. They're growing as white colonies because they've been mutated and they don't produce the pigment that the wild type was producing. So we didn't even need to do any sort of stamping. We didn't need to use different types of plates. You could just use nutrient agar plates, and you could expose your Serratia, you could incubate it up, and you would see some colonies pink and a good chunk of them that were white, right? We also could see, we leave those plates in the incubator for, days. These bacteria like room temperature, so they grow a little bit slower. And after a week, you could still see the pink and the white. But after we left them in there for maybe 2 weeks, a lot of those repair mechanisms that I just left that slide, those repair mechanisms kick into gear, and those white colonies go back to pink. They correct it, they fix it, and they produce the pigment. That's pretty stinking cool, I think, right? You could see the mutation, you could see it correcting over the course of time. Kind of cool, all right? What we do now is a little bit different. We take a couple of different types of bacteria, we put them onto the plate, we expose them to different times of UV light exposure, and then we incubate them up. Some bacteria are more sensitive to that than others, and we can see some variation in the growth patterns on those plates. We're not working with Serratia anymore and seeing the pigment change, but maybe something that we might think about throwing back in there, because I kind of think it's cool. So this gives us an opportunity to look for mutants, to see what sorts of things could be occurring. And in this case, like I said, we're using ultraviolet light to create these mutants, right? And an auxotroph is something that needs that little handout, if you will, to give it the ability to grow, right? So there's other ideas with this, thinking about mutants, all right, knowing that some things can cause mutations. And I showed the nitrous acid that was a potential for causing mutations, right? How do you know some substance is safe, right? You just wait for the FDA to tell you, right? How do you think they know if it's safe or not? They do something like this, all right? And there's potentially, although this seems good, there are some potential issues with this as they start testing specific things. They're testing things individually, right? But oftentimes you may buy some product and it's got handfuls of substances in there all acting together. They're not testing all of it as one unit. They test individual things. What happens when it's all mixed together? That might be a different story, right? That's beside the point, right? So similar idea with auxotrophic stuff. Something needs a little handout. So salmonella here, a bacteria that can cause some issues with your, intestines. Salmonella, we've got a mutant that requires histidine, right? So we already know we need to supplement these guys, right? And this Ames test is a test that's used to identify if some chemical agent, if some substance is potentially mutagenic, if it can alter the DNA, right? And in order to test for this, what they're really looking for is, can we cause a mutation that allows these cells, these salmonella, to now grow on media that doesn't have histidine? So it's a back mutation. It's making it go back to that wild type, the ability to grow with the nutrient not being in there, all right? So as we see this here, all right, we've got a control, and now we've got our mutagen that's coming into this mix, all right? Ames test is not something that I have done in the lab. And as I initially started looking at this, it made me go back to the book. I have heard of the Ames test. I've known a little bit about it, but it's not, as I got onto this a little bit before reviewing, I was like, what's up with the rat liver? Why is that in there? Right? Kind of odd, right? Was what I initially thought as I'm looking at it. And I'm like, well, this is our control plate and they're putting this in there. Is that serving as some sort of control stuff? No. Lots of things, chemical agents, maybe are fine until the liver has their way with them and tries to metabolize them. There's all sorts of enzymes within the liver that might potentially be acting on these things, right? So exposing them to some liver extract, those enzymes are potentially present. So now it's either a mutagen on its own or potentially a mutagen when it's been metabolized by the liver, right? So by giving it some extract from the liver, we've got both opportunities to really look at that. Is it the agent itself, or is it as it's been metabolized? You could probably set up a third experiment with no liver extract at all and really see, does it need to be metabolized first or not? I don't know if that matters, right? So this tube just gets the liver. The next tube gets the liver plus the potential mutagen, all right? There's gonna be some mutations that happen just spontaneously, just normal mutations that can occur. So these mutants that need the histidine, there could be a few of them that mutate on their own, all right? And here we can see a few natural revertants. There's two of them that show up on this plate that back mutated to the wild type. In this instance, with our potential mutagen, now you got a boatload of them growing on this plate, all right? So that indicates that whatever this possible mutagen is, truly, it is a mutagen because it's caused a lot more back mutations than just our simple control, all right? Lending to the recognition that this agent is mutagenic, right? And sometimes working maybe in a lab situation, you see all sorts of different chemicals. There's lots of safety mechanisms with material data sheets that spell out specific things. Are these hazardous? What do you need to do? Whatever it might be. Sometimes it labels it and says, this is a mutagen, right? You need to be careful because it's potentially cancer causing if you get exposed to it, all right? There are other things that they call teratogens. A teratogen is a mutagen, but it causes mutation in your sex cells, all right? That's more bad news, all right? That's something that you're gonna pass on to your offspring, all right? If you have offspring, all right? And maybe you don't want to, all right? Because it causes issues, all right? So different but similar sort of an idea, just depends on how it's mutating. Okay, shifting away from some of that. Our bacteria, as we've described them before, these are asexual organisms. They replicate their DNA and create a new, two new cells that are genetically identical. They're clones of themselves. And as we grow bacteria up on a petri dish and we see individual colonies of bacteria, it's the result of a single bacterium that replicated countless times to give you visible colonies, where there's millions of cells, right, that give you that one individual colony, all genetically identical, all right? But it's not always just straight up, this is all I got, all right? There is the potential for bacteria to pick up some variation, even though they're replicating asexually, all right? So it depends on the process, all right? All of these we think of as some horizontal gene transfer. So not from this E. coli to that E. coli, not love is in the air and there's bacterial sex, right? It's not like that, right? It's different mechanisms, although conjugation is kind of like that, but it's a little different, all right? So transformation is a process that, bacteria are really pretty good scavengers in their environment, pulling in things, and maybe some of the stuff they pull in happens to be some nucleotides. Maybe we could do something with that. If it's just a chunk of DNA, they'll likely just break it apart into the individual nucleotides and use those monomers to build more nucleotides. But if it happens to be like an intact gene that they pull in, maybe we could use that instead. We've found something, right? More than just a little piece of information, we've got a full set of instruction, and we could potentially put that into the genome. Sometimes we think more like plasmids, this little circular chromosome that might be inserted into a bacterium, maybe something we've done deliberately when we think biotechnology. Some bacteria we describe as being competent. They're able to do transformation. They're good scavengers and will pick things up. There are other bacteria that might be able to be manipulated, and we may make them competent. Maybe growing them in the presence of some calcium salts can cause their cells to be a little more porous and allow for the uptake of these of the DNA, whatever it might be, plasmid or chunk of DNA by itself, right? So transformation, uptake of naked DNA, all right, just pulling it in, all right, whatever it might be. And in this case, incorporating into the bacterial chromosome, giving it something that it didn't have before, all right? Transduction uses a virus, all right, a bacteriophage, with its DNA, or potentially in this case with transduction, with the host that it was leaving from. It's got the host chromosome, or a chunk really of the host chromosomal DNA incorporated into the capsid or the phage head, and it gets inoculated into the bacterium, right? Sometimes we see a bit of both, a bit of phage DNA and bacterial DNA, either way, all right? And it can incorporate into the chromosome, all right? So transduction uses a virus to infect it with hybrid DNA. Conjugation uses a plasmid. So one bacterial cell has this plasmid, like this guy over here, right? It's got this plasmid, but this plasmid is something that allows for the contact through a pilus, the contact of its cell with another cell in close proximity, all right? It causes this conjugation pilus or tube to form in between both of these cells, and it copies and shoots a replicated copy of that plasmid to the cell, all right? And now we've got a cell that didn't have it that does. Antibiotic resistance oftentimes we think about it being transferred from one type of bacterium that's got that resistance gene carried on a plasmid, transported into a different type of bacteria that never had the resistance, and now they do, right? And they're able to share it with others as well, right? So 3 different ways for horizontal gene transfer to bring novel nucleotides into some of these cells, giving them stuff they didn't have before, right? Potential advantage for sure. And here we can see our Vibrio cholerae, that this is through an electron micrograph. Another one that with some of these processes able to share some of these virulence factors, cholera toxins, with some others. So here we can see conjugation with the cell that's got this plasmid, all right? And there's a boatload of different types of plasmids, all right? This F plasmid is one that is going to cause conjugation between these cells, and we could potentially have other plasmid in here as well, and maybe in the process of this conjugation we might pipe in some other stuff as well, all right? The cell that's got the plasmid, we call it an F plus. The cell that doesn't is an F negative, all right? And the plus ones want to make the negatives positive, all right? They would like to share these plasmids, all right? So coming in proximity with those that don't have it, and it doesn't have to be the same species of bacteria, all right? Could be somebody different, all right? And the pilus forms, and like I said, replicating it and shipping it over. So now we've got multiple positive cells, all right? Sometimes when this occurs, we wind up incorporating, or maybe the bacteria that's got the plus cell, maybe it's starting its replication, and we're also starting some conjugation, right? And sometimes we wind up grabbing some of the host chromosome and shipping that into that negative cell. And that gives it the opportunity, instead of staying localized outside of the chromosome, it incorporates right into the bacterial chromosome, right? We call those guys HFR cells, high frequency of replication, right? So we also sometimes say these cells, they're promiscuous, all right? They would like to do it any chance they can get, all right? If we would call conjugation doing it, all right? So every time this bacteria that's now got this plasmid incorporated into its chromosome, every time that it replicates, and it could be every 20 minutes, it's going to basically read through that plasmid chunk of DNA that says, hey, I ought to conjugate too. And so the normal F plus cell does it just randomly, may bump into somebody and conjugation may happen. But with these HFR cells, it wants to happen all of the time. And if we're talking drug resistance, antibiotic resistance, a situation like this can spread very rapidly through a population of different types of microbes that never had it, right? That's the idea. So that's what we're seeing here. And in this case, this lac gene, the ability to grow in the sugar lactose, we're not looking at antibiotic resistance, but similar idea could definitely be a possibility with that. Okay. So this basically just showing, and I'll quit right with this, as it integrates in handful of different genes, and as we're looking at the specific plasmid stuff, some other genes within there, and dependent upon how much of it or all of it that incorporates in, it gives variation to these respective cells. We also think about, the host chromosome as well as we're coming in there, right? But this kind of mapping it out as far as what we've got, and this is a time. So at about 100 minutes into it, we could get every single segment of this replicated and potentially shipped over. And just depends on those circumstances as far as what they're donating, what they're piping over in the first place. All right, we will pick up super close to the end of this. We'll pick up with a little bit of Jumpin' Jeans on Monday and then start with chapter 12. Have a great weekend, everybody.